right, bacterial. So we talked yesterday about regular DNA in a eukaryote and how it replicated itself. One side, called the leading strand, was copied in order. The other side, called the lagging strand, was copied in little pieces. What were the pieces called? Right, Okazaki fragments. And then they had to be sealed together with an enzyme called ligase that hooked everything together at the end. So this is not an issue in bacteria because bacteria have a loop of DNA, if you recall. It's not a linear DNA. So in other words, whereas a eukaryotic cell, if it opens a linear DNA, one side's going to be 5 to 3 and the other side's going to be 3 to 5, and it can only copy in one direction. This is not going to be an issue because if the DNA is a circle, as soon as that circle starts to open up, we imagine this is our DNA, and as soon as this starts to separate, it can basically copy in both directions. This one can copy this way, this piece can copy that way, and each one will just copy in a loop. So there's not going to be any problem with Okazaki fragments and having to seal anything together. It'll basically start at the origin of replication, and it'll make an exact copy. So bacterial DNA, much, much simpler, easier to replicate, and this is why I mentioned way back when we talked about mitosis and meiosis that it takes like 13 hours for a cell to go through the cell cycle in a eukaryotic cell. Because it's got to go through S when it replicates the DNA, and then it's got to go through mitosis, which is a complicated process. A bacteria can go through an entire cell cycle, copy its DNA, and divide in about 20 minutes. So bacteria are much, much faster. Oh, sorry, does anybody still need that information? So it can copy in both directions, no Okazaki fragments, very, very simple. This is a picture, again, just showing how the bacteria can copy each side of the DNA. They just copy in opposite directions. Technically, eukaryotic DNA doesn't unzip at one end. I showed it that way to simplify it. It actually copies in several locations at once. It makes these little replication bubbles. But honestly, you don't need to understand the replication bubbles. This is showing what it does. You still just understand how it can only copy in one direction. That's all you really need to understand. Which brings us to the last thing. And that is, because the DNA is linear, the tips of the DNA are going to have a problem getting copied. Because, if you remember, when I showed this unzipping, this piece of DNA had a primer that attached here. And then the new strand was growing. Eventually, this primer is going to leave because it's made out of RNA. I told you it's sort of like the bottom of a jacket that you hook a jacket into to zip it up. When the primer leaves, you're going to be left with this little segment of DNA that has not been copied. Everything else has been copied, but the problem is you can't copy here in the 3 to 5 direction. It can't build this way. So every single time the DNA makes a copy of itself, the tips of the chromosomes, the tips of the DNA, are not going to be able to be copied. And therefore, DNA is going to get shorter and shorter with each replication. And this is what limits, from what a lot of what we understand today, this is what limits how many times a cell can copy itself. Cells will only copy themselves for so long because the DNA gets shorter and shorter and shorter. Now, there would be concern if all the important genes ran all the way to the tip of the chromosomes. Because if the important genes went all the way to the end, that would mean every time DNA copies itself, you would lose important information off of your chromosomes. Luckily, that does not happen. Yes? This does happen to us. This is exactly what's happening in your cells right now. Your DNA is getting shorter and shorter every time it gets copied. And eventually, this is part of our aging process. This is one of the reasons why we die, why we age. Now, here's the thing, though. You don't lose important information, and then I'll answer your question. At the ends of your chromosomes, you have what are called telomeres. Telomeres are sort of these repeating sequences of DNA that's just junk. So even though your DNA gets shorter every time your chromosomes copy themselves, we know that there's like this extra DNA there. Specifically, it's sort of like putting bumpers on the edge of a table. So when you bump against it, you might scrape something, but you don't lose anything important. That's basically what's happening. They compare them kind of like the little aglet things on the tips of your shoelaces, even though that sort of protects them. So even if my DNA gets shorter, if all of this DNA at the end here is all junk anyway, this can copy itself and lose a piece, copy itself again and lose a piece, copy itself again and lose a piece. The pieces that are lost are just junk. They're just there specifically to protect the important genes. But this is a part of our aging process. We know for a fact that eventually when these telomeres get length, it programs the cell to never reproduce again, and some cells will actually go through a programmed cell death when the telomeres get too short. So one of the things they're studying is the enzyme telomerase. Telomerase is an enzyme that actually makes the telomeres longer again. It sort of restarts them. The only cells that we see telomerase normally in, there's only a few. Tumor cells have it. That's why cancer cells don't just reproduce until they die. They just keep reproducing and they never die. Technically, based on this, they should only be able to reproduce a certain number of times, and then their telomeres would get too short and they would die. But this enzyme re-lengthens the telomeres so that it basically restarts each time. When this says germ cells, it doesn't mean germs like viruses. It means germ cells is another word for gametes. So we also know that sperm and eggs have telomerase and the telomeres restart. So you don't start off life with shortened telomeres. One of the things we know that happened with Dolly the sheep, she was the first cloned mammal in 1996. Dolly the sheep had a lot of old age diseases when she was very young. For example, she had arthritis as a young sheep. But she was cloned from an adult sheep cell that was 6 years old. The adult sheep was 6 years old. So we think one of the reasons maybe that she had some of these old age problems even though she was young is because she was cloned from a cell that already had shortened telomeres. She was cloned from a cell that was already technically an older cell. Yes?
When you clone something, does it have the same personality?
No. Personality, from everything that we understand, has a ton to do with what DNA, it has a genetic component, but it has a ton to do with how that DNA gets influenced by the way you're brought up. So your personality, although there may be parts of your personality that are genetically programmed, there's a huge range of how you're going to turn out based on everything that happens around you once you're born.
Okay.
So yeah. So anyway, so that's where a lot of studies are right now, is looking into telomerase and telomeres. You can actually go get genetically tested to see how long your telomeres are. It'll give you a genetic age that you could compare to your actual age. And so you could go in and I could be 48. I could go in and find out that my telomere genetic age is 60. Maybe because I haven't taken care of myself, so the more damage I do to my body, the more my cells have to reproduce, the shorter my telomeres are going to get. And it wouldn't necessarily mean that I'm going to die sooner, but it would definitely be an indicator that my body is aging faster. And so supposedly, there are things you can do that will slow down how quickly your telomeres shorten. I think a lot of the things on the market are probably scams, aimed at scaring you into thinking if you take this pill or whatever. Because I don't know that you can re-lengthen them, but you can definitely slow down how quickly they get shorter. All right, so that's everything in the first part of the chapter. Before we move on, I wanted to